Microphone on. All right. All right. Shit, make sure my pencil works. No. Why again is the pencil not working? Come on. Okay, JJ, maybe this isn't going to work because if the pencil doesn't work. Yeah, right. In one more second. Thought I had it all perfectly laid out. Pencil's at 99%. Sure, let's. Got it. Okay, here we go. Chapter 9. Okay, so a couple things before I dive into chapter 9. Some of it is structure related. So when it comes to asking questions on the exam, I am not going to have structure related questions. Like, I'm not going to ask you, where's the insula? Which bone is this? Things like that. That's anatomy, and you can take that in when you take your anatomy course. So I will try to focus specifically on function, though I'll have to refer to structure so we know where we're at in the brain. Okay. All right. So it starts off with emergent properties. This means, like, for our brain, one neuron, just a singular neuron, does nothing for us as far as we think of cognitive behavior, movement, any of those things. You have to put the whole system together to get movement, to get feelings, things like that. So individually, it does not work. It has to function as a whole. Evolution of the nervous system. Done. We are not going to study the evolution of the nervous system. Therefore, I'm not going to cover this image, but yeah, sure, we can see that here we have a network, and we're slowly building up to maybe something that's getting more brain-like structure, little tiny fish brain, bird brain, and then finally you see we have our brain. But again, I'm not going to ask you questions about that, but yes, over evolution, it has evolved. Now we really start into the good stuff. Let's start with, do I ever start with Mallory? Mallory, do I ever start with you? You're like, yes, all the time. There are germ layers. Can you name one germ layer? And again, you can pass. Just go pass. So these are our embryonic layers. It's fine. Jade, any chance you got one? You're okay? Go to JJ. Endoderm is one of our germ layers. Daquan. Mesoderm or mesoderm. Correct. Michaela? That's fine. Ectoderm. Okay. So as the human fetus starts to develop, there are three main layers called our germ layers: endoderm, mesoderm, ectoderm, that through the process of growing turn into our different physiological systems. Now, I didn't mention this in the endocrine system because the endocrine system evolves from all three. Some of it is brain related, some of it is singular cell related, digestive system related, so it really does pull from all of the three germ layers. But when it comes to the nervous system comes from the ectoderm. So we'll write nervous system down there. Lexi. here's your question. Any chance you know what other system comes from the ectoderm? And here's my clue. Think about if you were an insect. Ectoderm. And we think about an insect and its exoskeleton. What is the exoskeleton of an insect? And if that makes no sense, you can go, I've lost you and I have no idea what you're talking about.
Like skin.
Nailed it. See, you knew it. So the two main systems to come out of the ectoderm are the nervous system and skin, which is, I think, entirely crazy to think our skin is the same origin as our brain, because they massively different. As we go through the other systems, we'll learn where the other layers play a role. In terms of what do we need to know about nervous system development? We need to understand, one, it's ectoderm derived. So as we're a ball of cells and ectoderm is growing on the outside, eventually what we'll get is something called a neural plate. I believe on the next slide, there'll be a neural plate. This thing is your neural plate. So that's all ectoderm type tissue. The next couple of things that it's going to show are what then develops. So our neural plate will, let's see if there's a piece of paper. Here's what our neural plate does. Maybe. Consider this all the ectoderm. And all we do is we fold our ectoderm like this, and now we're starting to create our neural plate. And eventually this will grow into our central nervous system. So we go from neural plate into a neural tube. So let's look at that picture. There's our neural plate, and then we kind of fold it up on itself, and the inside of this becomes the neural tube. And then like a day later, everything's fully there, we can say that the neural tube becomes our central nervous system. And then there's this tiny thing above it called the neural crest, which becomes our peripheral nervous system. Yes?
So would synonyms be like the notochord and the dorsal hollow nerve cord?
Say that again?
Would would it be saying the same thing using those two words?
I don't want to put other lower vertebrates into the same setup. So, okay, so here's what you remember. You remember that I've got your ectoderm folded up. Now I've created first we got that kind of divot, that's the neural crest, the neural plate. Up here would be the neural crest, fold it some more. Now I'm creating the neural tube, which is our central nervous system, brain, spinal cord, and then the little thing on top would be your peripheral nervous system. All right, so that's the first days of development. Let's see what else it says. All right. Then as we continue on, I'm not going to be asking you what day does this happen or what week does this happen. I just want you to know the steps involved, the making of these different areas. So roughly a couple days later, we start to separate that neural tube into different portions of the brain. So the first things to come out would be the forebrain, the midbrain, and the hindbrain. So we go from neural tube to a forebrain, a midbrain, and a hindbrain. And then a couple weeks later, we start to develop more. And you will need to remember that the forebrain will turn into the cerebrum and the diencephalon. The midbrain just remains midbrain, so that's an easy one to remember. Midbrain just stays midbrain. But the hindbrain is going to develop more into the cerebellum, the pons, and the medulla oblongata. We'll get into what these ventricles are a little bit later. I more want to focus on neural tube to the forebrain, midbrain, hindbrain, then to the development of the forebrain to the cerebrum and the diencephalon, and then the hindbrain to the cerebellum, pons, medulla oblongata. It would be great if, as you start studying for this exam, that you can actually draw out how, where these structures start being created. So for instance, you could go ectoderm will lead to the neural crest, but also the neural tube. And from the neural tube, we get three things, being the forebrain, the mid, and the hind. And then from there, you would start to separate it out even more. So mid stays mid, hindbrain turns into the cerebellum. I'm not going to write these all out. And then the forebrain becomes that cerebrum and diencephalon. And we kind of basically just say, good, let's stop there. We've basically created most of the structures that we need. Okay. It's just showing you pictures on how, again, we're seeing that development. So you see forebrain, midbrain, hindbrain. You can see how the forebrain is going to break off into pieces, how we get the hindbrain to become the medulla oblongata and the cerebellum and the pons. But I'm not going to have a picture like this, and I'm not going to point to this thing and go, what is that? Nope. It is neat to see how fast the eyeballs develop. Week 11, and you've got something that looks like an eyeball. I think I will order us some zebrafish eggs, and we can watch their development over the time of a week. Because they're very translucent, the eggs are clear, and then the actual embryo is clear, and you can start to see their eyes will form really big, and then you can also see the pulsating of the blood through the clear fish as it develops. It's really cool. So if I can remember, I'll order zebrafish eggs. All right, so it keeps on going. Now we've got some type of directional system set up. I'm not worried about that. It's just showing you development. This is just showing us here are the portions of the central nervous system. We've got the brain and the spinal cord, and the spinal cord you can then split up into different sets of nerves. So we've got the cervical spinal nerves, which will function to help the neck, lower part, like all the muscles that are right here, the neck and the shoulder muscles. So that will help with movement, but also sensory. Then we could go to thoracic. We're talking more about visceral functions, digestive tract functions. And as you move down, it'll control functions that are lower. Okay. So you should know that, yeah, I know my central nervous system is made up of my brain and my spinal cord. And then the different parts of the spinal cord would be spinal nerves, thoracic nerves, lumbar, and sacral. Oh no. What do they look like? When you look at a cross-section, or so a cross-section either this way at our vertebrae, or a cross-section this way or this way for the brain, we're going to see two different colors that are going to show up. It's either going to be gray or darkish yellow. I think it looks more darkish yellow, maybe like egg yolky. Versus white matter. Okay. So for the central nervous system, the gray matter is lacking the whiteness because it's not myelinated. Remember, myelination is like a lipid-based substance. So if you think of fat, it's usually this lighter type color. So gray matter is unmyelinated. And it's these, I'm going to draw some neurons. So that's the dendrite, that's the axon, the axon and the terminals. They call things that are clusters of these nuclei all together, because that's how they're going to kind of be. This thing is just called a nuclei. A cluster of cell bodies in the central nervous system are called nuclei. It's just a vocab terminology. When it comes to white matter, again, white matter is myelination. So when we have clusters of these together, we are going to say that they're axon bundles because that's where your myelination is occurring. And if it's in the central nervous system, we call them tracts. So gray matter lacks myelination, darker in color, and what we are visualizing is a cluster of the cell bodies together, and we call that nuclei. White matter has myelination. What has myelination? It's the axons. So when we get a lot of axons together, we call those tracts. Just terminology for this. This is a cross-section of the spinal cord. So there's a number of things to point out in this slide. You can see that we've got gray matter more on the inside. White matter is more on the outside. Okay. Then it's going to point out that these spinal nerves, one is coming from the back and one is coming from the front. That's going to play a role in sensory versus motor. So some of these nerves coming into the spinal cord will be sensory, and then some of the nerves coming out of the spinal cord will be motor. Makes sense. Then there are these things called the meninges. These are just the layers around the spinal cord, and they are the same layers for the brain. Okay, so we got pia mater, which is the closest to the spinal cord, and arachnoid matter, which is the middle, and then the dura mater, which actually looks like plastic. And when you get into comparative, or if you take anatomy, you have to cut off the dura mater, and you have to physically put your scissors into it and cut it out, like you were cutting open a plastic package that you got at the store. You know, the really plastic packages that are hard to open that you slice your fingers on? That's like the dura mater for our spinal cord. Okay. Things you know. You know that the brain is enclosed by the skull or the cranium. You know that the spinal cord runs down our vertebral column and that there are bones protecting it. I'm just going to go back to the slide. You see that here's the bone, and in the middle of the bone is the spinal column. So what it's doing is it is trying to protect from the front and the back our spinal cord. If you could touch your spinal cord, that would be horrendously bad. There are conditions when your spinal cord or the fluid of your spinal cord is born on the outside of you. That's spina bifida. So we protect it by putting it in the middle of the vertebrae. And then we just talked about the 3 layers. And I'll talk a little about it more when we hit the brain. Here we go. Brain. This is the bone, the cranium, so we're thinking more of the things we can knock on your cranium. Then I'm going to start down with the brain. This tiny layer, you really can't see it, is the pia mater. It's so, when you look at a brain and you put your hand, I don't know how many, just put your hand on a brain, that top layer you're touching, that's a little thin layer of tissue, that's pia mater. These fibers we're seeing, all these white little fibers, that's the arachnoid matter, and you can guess that it gets its name from arachnids, spiders. It's not that spiders are in our brains, it just looks like a spider web, a cobweb. So that's why they call it arachnoid, or the arachnoid membrane matter. But there's a lot of space here, so what we're gonna put in this space is our cerebral spinal fluid. That's why that space exists. The reason we have these layers set up, one of them is so we have that space to have cerebral spinal fluid flowing around the brain. And then the dura mater is the tougher thing. Again, it's like a white layer of plastic on top of it. And then you have your bone. Another interesting thing to point out is that our brain has what's known as sinuses. So sinuses serve two purposes. One, our brain is heavy. So if we can create some open pockets, it's going to help it be lighter and have a little bit more ability to float. The other reason the sinus is there, there's these structure. Now I'm writing blue on blue. This structure right here, that's called a villi. And it's basically all the cerebral spinal fluid that's flowing around. We don't want it always to be flowing around because it will have taken up waste. So we got to somehow figure out how to get old cerebral spinal fluid out. And it'll just come out into these sinuses, and we can recycle it or get rid of it through the cardiovascular system. Okay, so we're protecting our brain, and we're making sure it has the right environment. It's got cerebral spinal fluid going around it. That's what those different layers can do. So, cerebral spinal fluid, why do we care? First, it's a salty solution. Salty. So we got lots of ions floating in it, the electrolytes that we need, sodium, chloride, potassium, salty stuff. That's going to help us continue on neuroprocessing so that we can have action potentials, things like that. So that's why we care about cerebral spinal fluid. That's one of the reasons. All right, so who makes the cerebral spinal fluid? I did tell you at one time on chapter 8 that the epididymal, I'm going to spell this wrong. Hold on, let me make sure. It should be. Is it going to show me? Hold on. Nope, it's not going to. Okay. Anyone remember how to spell epididymal? All right, I'm at least saying it. Epididymal, those cells. We had the astrocytes, we had the epididymal cells, we had the oligodendrocytes, Schwann cells, all of those. So the epididymal cells are going to help make that cerebral spinal fluid. They are going to be found, and what you're going to find in an area that makes the cerebral spinal fluid, is a thing called the choroid plexus. So what I'm going to do now is I'm going to skip to this figure and show you how this is all done. We have a couple choroid plexuses to note. You can see one is right here. You see that red type substance? That's the choroid plexus. It's found within our third ventricle. And the epididymal cells there are going to make a lot of cerebral spinal fluid, and then it will just join the open spaces that flow around the brain. Another place we can make cerebral spinal fluid is the choroid plexus, so this red area right there. And that's found in the fourth ventricle, and that will just add to it and have it flow around the brain. So you've got third ventricle, choroid plexuses making cerebral spinal fluid. You've got fourth ventricle, that choroid plexus making cerebral spinal fluid, and then it is just flowing down and around and up and through. But eventually we're going to need to remove it through the arachnoid villi because you don't want used cerebral spinal fluid to continue flow around your brain because it's just going to have a lot of waste in it. So we got to get rid of it. That's how you spell epididymal. Okay, I knew it was there somewhere.
So how does the waste get filtered through the?
Yeah, so it's going to get sucked into the villi, and then the veins that are found up emptying into the villi or in the epidermal space will just pull it out. So let's look at what we're seeing with these cells. I've got these epididymal cells that are right next to a capillary, and it is selecting what should become part of our cerebrospinal fluid. So it's not everything coming from that capillary that will become cerebrospinal fluid. We're selecting certain things. The epididymal cells figures it out and starts producing the cerebrospinal fluid. Okay, let me go back. Okay, so how we're gonna move things from one place to another? The osmotic gradient. That's what our body cares about. This is why we did that lab, because there are so many physiological systems that use osmosis to function. Other functions of the cerebrospinal fluid. So we got all the electrolytes that are going to help balance and keep that resting membrane potential and the ability to fire off an action potential. So that's the chemical typey thing. We've also got physical protection. So there's a place, the Dead Sea? I don't know. There's some place that has so much salt that you just float on top of it. You don't even need to do anything, and you're floating. That's kind of what the setup is here. Super salty so that our brain floats. The back part of our brain has the medulla oblongata, the pons, and these are our systems that keep us alive. Breathing, heart rate, vomiting if something is bad in us. These are the core things for survival. And if we didn't have this buoyancy created by our cerebrospinal fluid, our brain would just smash into the back of our occipital lobe, and your pons and medulla oblongata would get smashed. I think the thing you would die of, that we always die of, is suffocation, because breathing is monitored by all of this. So, great. We have cerebrospinal fluid, helps keep our brain a little buoyant so it doesn't smash. Also, everyone has run into something with their head at some point. It's a matter of life, we run into things, or people run into us. We have the cranium right here, which is really hard, and if we didn't have cerebrospinal fluid, even just the mere small portion of it, our brain would slam into our cranium, and you would have brain damage. We still can get brain damage. We know that through concussions of football players or other sports. But at least in general life, when we're not ramming our heads into other people, this helps protect us. This is another image of those ventricles. Again, they are open spaces in our brain to help keep that buoyancy, but also they have those core plexuses that are going to keep making cerebrospinal fluid. All right, so we did that already. This is that villi that is going to kind of collect some of that cerebrospinal fluid, and then it moves back into our bloodstream through veins. All right. Now, another key part. If I close this, does this mess me up? Oh, no. Uh-oh. I messed up. Sorry. All right, blood-brain barrier. Super important to protect what gets into our brain. So the astrocytes are the cells that are going to create a very tight barrier. This is your blood-brain barrier to control what's going to get into our brain, our cerebrospinal fluid, or just other areas. So toxic chemicals, anything toxic, it's going to try to not allow in. Things that can pass through are lipid-soluble molecules. You know this already, that lipid-soluble molecules get through the cell membrane because steroids are lipids that go through the cell membrane without needing a receptor. The receptors are the inside of the cell. I don't know where I left off. I think, Mallory, it's back to you. How would the blood-brain barrier work against you? I feel like we talked about this already, but if we didn't, whatever. It's a wonderful system, and I'm so glad we have it, but there are cases where it does not help us. Want to pass? Totally fine. All right. Jade? JJ?
Well, you can't get treatment through.
There you go. So if you got a brain tumor, it is harder to treat brain cancer because of the blood-brain barrier. It's going to think it's a toxin. And so you really have to manipulate. This is where we need those chemists to figure out, how can I take this chemotherapy drug and change it in such a way that it actually will cross the blood-brain barrier, and now we can go attack that cancer. Okay, so it's great, but then not so great if we need to help the brain. This should be a picture of it. Here's what I want to show you, and I need to show you this because there's something that we've never seen before. That's a capillary, right? That thing's a capillary. But how do you make a capillary? To us, it's just this tiny tube. That's a capillary. But what is it really made of? That's a nucleus. That's a nucleus. So what they are endothelial cells that are just linked together to make a tube. You have to start to memorize what endothelial cells are, because a lot of our body's functions are the passing off of information between capillaries. So we have to understand capillary tubes are made by just stringing endothelial cells together. Okay.
What's the difference between endothelial and
Epithelial is this.
okay.
Yeah, endothelial is that. So epithelial dies. Like the origin, I don't know the answer to. And I think that actually gets to the heart of how they're different. All right, so endothelial is going to make your capillary tube. And what sits on top of the endothelial is like a tiny layer of connective tissue, super tiny, just to keep things together. And then you see these little things called pericytes. They're like little feet that have just wrapped around the capillary. They've connected themselves on top of the capillary. Those little feet processes come from astrocytes. So you can see here an astrocyte and multiple astrocytes wrapping their little feet around the endothelial cells, that capillary tube, and that creates these tight junctions to not allow anything through. It's not going to leak out. It'll get through, but it's going to be controlled by the pericytes and the endothelial cells to decide what should get through. We should not have any leaking happen. We're going to go through the cells. We're not going to go in between the cells. Through the cells, you have a lot of control, channels, things like that. In between the cells is just leakiness.
So is the pericyte the foot of the astrocyte?
Yep, you got it. We have the same idea set up in our bladder, that you have these tight junctions, and these tight junctions are absolutely necessary, because if you didn't have them, then your urine would just leak out of your bladder into your pelvic cavity, and that would be gross. So we're happy tight junctions exist. All right. What does our brain need to survive? It needs oxygen and glucose. There are reports that our brain can survive off fat too. Fat can be turned into a energy source, but for the most part, we'll focus on glucose. Oxygen easily passes through the blood-brain barrier. Glucose does not, but we already know that there are these transport, GLUT4, GLUT2, whatever, that will move glucose through. And if we are low on glucose, which is hypoglycemia, everyone has hit a moment of hypoglycemia in their lifetime. You can feel shaky, you can feel lightheaded, you may start sweating, and worst case scenario is you pass out and go into a coma and die. So just like if you're diabetic, this is a big issue because if you're diabetic, especially type 1, your insulin isn't going to bring in glucose into your cells because you're not making insulin. So a diabetic takes an insulin shot. Doesn't go in there. I don't know why I'm doing this. Takes an insulin shot, and then that removes the glucose. It pulls it into all the cells. Insulin's working, and it's pulling into all the cells. But you overdid it. If you overdid how much glucose or how much insulin you gave yourself, you can turn into hypoglycemic. So nowadays, diabetics, type 1 mainly, although type 2 too, have these patches on their arms. There are a couple students that were in last year. They have these patches on their arms, and they're called Dexcom receivers. My husband's type 1. And it just samples how high is your blood glucose. And if it's really high, then a pump that is connected somewhere else down here shoots insulin into your body. Then we start removing the glucose from the blood, but sometimes it overpumps. Or if you're doing it yourself and you inject, you overdo it. You make a mistake on how many carbs you took. So that means it removed way too much and utilized too much, and you fall into hypoglycemic. And then what you start to hear, anyone who's type 1 and they have the setup, they start beeping warnings. Beep, beep, beep. Beep, beep, beep. And you're just like, what is that? And then you realize, oh, you just need to get some fruit or drink something that is sugary, and then everything corrects itself. Spinal cord. Okay, now we're done with all that fun stuff. We're going into our spinal cord. So I said that there's a passage of nerves to go into the spinal cord, and there's a passage of nerves that come out of the spinal cord. So, into the spinal cord, we have the dorsal root ganglia. And we say they are afferent. Anything afferent is another fancy term for sensory. So this makes sense that if it's going into the spinal cord, it is a signal that we have to, coming from somewhere else in our body, coming into our spinal cord, that will go up to the brain. So it comes in through the back of the spinal cord and then goes up to the brain. So dorsal, always sensory, fancy term for sensory is afferent. Then the ventral. The ventral are afferent, and sorry, nope, I meant to put it there. So sorry. The ventral are going out of, and they're the ones that are carrying motor information. So it's coming from your brain. Brain's telling you need to write something down. Goes down the spinal cord, out the thoracic, and now you are writing info down. We should see. Let me get to the picture first. Okay. So this is an example of dorsal. That's dorsal. And then this is ventral. Same thing on the other side. Ventral, and then this is dorsal. All you have to remember, ventral, motor, coming from the brain down to a muscle or a gland, something like that. Dorsal, coming from somewhere else of your body, going into the spinal cord and going up to the brain. Afferent. There are also, we're seeing in this slide, you're seeing the gray matter, which is more inside. But then we have all these funny colors all around. These aren't separate colors. They are clusters of tracts of neurons. Remember, tracts of neurons are clusters of axons. And each one of these different colors is going to carry different information. Don't memorize everything in this slide, but understand tracks. If they are ascending tracks, so it's saying all of these are ascending tracks, clusters of neurons, and it is sending info up to the brain. If it is a descending tract cluster of axons, then it is coming down and is going to do a motor function. Okay, so that means I want to go back to this side. Gray matter, not particularly. What was I thinking? So what it's going to control is visceral and somatic sensory. If it's the dorsal one. If it's dorsal. Dorsal we already know to be sensory. So what is visceral? A visceral reaction is like a gut reaction. So just think visceral, think gut. Somatic sensory is a little bit odder. It's skin and muscle, things like that. Okay, so we're either going to receive information from our gut telling us maybe we have a tummy ache or we're hungry, something like that. Or we're going to receive information from our skin or our muscles or our joints to tell us stuff about that. Then I'm going to focus, so then we've got a lateral, but I kind of put it also in ventral. You got motor nuclei. Visceral motor nuclei is the gut moving. How am I controlling all my digestion? Visceral motor nuclei would help with that. Somatic motor nuclei would be, again, referring to the skin, muscle, joints. Okay. White matter. Ascending tracts, we already just said, bring sensory info to the brain. Descending tracts are motor signals from the brain. There are these things called propriospinal tracts that do not leave the spinal cord. They're like in between. And then we'll get to what a spinal reflex is in a second because there's an image. All right, spinal reflexes. Who do I want to ask? Jade. Jade. Pretend this is a flame. And you're going to put your hand on the flame for like a second. Put it on. All right. Now, let's say I made the flame bigger and you were about to put your hand on it and it's super hot. Yeah. Do the motion of what you would do if you put your hand over the flame and it was super hot.
Put it over?
Yeah. All right, but what if I said you had to go low? Then what do you do? Right. Yeah. So this movement, see what she moved it back, but she also moved it back like this. She didn't move it back like this. It was always away, not above. That's a reflex. It's always done the same way through every person, no matter what you do. Same thing like when your doctor does this, whatever, patellar reflex. We always, when it's activated, goes forward. Doesn't go off to the side, doesn't go back. So reflex just means it's the same way every single time. For our spinal reflexes, something happens, info comes in dorsal, right, into an afferent, but it is integrated right into the spinal cord. This pulling away of your hand happens here. The idea of the fact that it's hot and you just got burnt or it's just super hot goes up to the brain. So yeah, we're going to send info up to the brain, but that actually gets processed after you remove your hand. Your hand moved off first before your brain goes, oh damn, that was hot. Now it feels like the same, but it wasn't. So a reflex for spinal reflexes is something that is integrated completely within the spinal cord for it to occur. All right, this is, I'm just gonna put this here. Why is this not working? Oh, because it's the wrong one. Ha, extra credit exam 2. We're gonna have to end there. So extra credit exam 2 will be the spinal nerves. We'll get into that next time. So let me shut this off.